Hi, my name is Tatiana. I'm a postdoc at the Center for Evolutionary Hologenomics at the Globe Institute here in Copenhagen. To me, hologenomics is the integrated study of the genome of a host organism along with the metagenomes of the microorganisms associated with the host in order to study kind of genetic interactions of the host and its microorganisms and the implications of these interactions. I got started with hologenomics because of the research I did during my PhD. Part of my PhD research focused on studying the genomes of Greenland sled dogs that are thought to be highly adapted to the harsh Arctic environment and lifestyle after having lived there for about 1,000 years. This led me to start thinking about how it's not only the genome of the dog that's adapted to the Arctic, but it's also the microbes living inside the dogs or on the dogs that help with a variety of different adaptations. So without examining these, we can't really fully understand the survival and success of dogs in this environment. As I mentioned, my PhD involved the study of Greenland sled dogs, and my current research is a follow-up on some of the hologenome-related questions that were sparked during the course of that research. I am currently studying the metagenomic and genomic data sequenced from oral swabs in order to characterize the oral microbiome and the host genome of dog populations from across Greenland. There are multiple different questions that we're aiming to answer relating to the composition of the oral microbiome, the presence of pathogens, including zoonotic diseases, and to look at differences between individual dogs, dog teams, and locations that the dogs live in. To me, I think this is a really interesting concept in general, looking at dog hologenomics, but in particular, I think the Greenland sled dog are a really poignant population to examine due to their unique lifestyle, dietary habits, and of course the changing environment in the Arctic, which makes it a really critical time for this research to take place. Interestingly, despite extensive behavioral, clinical, and genetic studies done on dogs, very little research has actually been done on their microbiomes. So whatever research that has been done has actually been more related to 16S data. So we are taking an approach using shotgun sequencing data, which will allow us to obtain information from both the host and from the microbiome. And that will potentially give us kind of a greater breadth of data to be able to address the questions that we're looking at. And on top of that, very little of the research that's been done has linked the two microbiome and genomic data together. So again, that will be something novel for us to be looking at. A fun fact about me is that even though I studied Arctic dogs from Siberia and Greenland for my PhD, and I even went to Greenland 6 times, the first time I actually went on a dog sled was in Sweden, so not so much in Greenland or Siberia, and it was even after I started my PhD. But I guess that's the downside of going to Greenland in the summer and the autumn for the first couple of